Dear students, Assalamu alaikum. In the last lecture, we studied about the humoral immunity. In this lecture, we'll study about the cell-mediated immunity. Upon exposure to a proper antigen, the T lymphocytes of a specific lymphocyte clone divide and release large number of activated specifically reacting T cells. A difference is that instead of releasing antibodies, whole activated T cells are formed and released into the lymph. Also, T lymphocyte memory cells are formed in the same way that B memory cells are formed in the antibody system. In case of the T lymphocytes, when it is activated by an antigen, many of the newly formed lymphocytes are preserved in the lymphoid tissue to become additional T lymphocytes of that specific clone. Now, to understand the cell-mediated immunity, it's important to understand the function of the antigen-presenting cells. Now, although B lymphocytes can recognize intact antigens, the T lymphocytes respond to antigens only when they are bound to specific molecules called MHC proteins. Now, these specific molecules, the MHC proteins, are present on the surface of antigen presenting cells in the lymphoid tissue. The three major types of antigen presenting cells called the macrophages, B lymphocytes, and dendritic cells. Now, the interaction of cell adhesion proteins is necessary for T cells to bind to the antigen presenting cells. Now, let's pause here to understand what are MHC proteins. The MHC proteins, as we just learned earlier, that the MHC proteins are present on the surface of the cell, so they are encoded by a large group of genes called the major histocompatibility complex. Now, after being expressed by the expression of these genes, they are placed on the surface of the antigen presenting cells. Now, the function of the MHC proteins is to bind the peptide fragments of the antigen proteins, which has earlier been digested by the antigen presenting cell. So, there are two types of MHC proteins. MHC proteins 1, which present antigens to the cytotoxic T cells, and MHC2 proteins, which present antigens to the T helper cells. Now, in this figure, the MHC proteins are shown on the surface of an antigen presenting cell, and there's a foreign protein which is bound to the MHC protein. Now this antigen presenting cell, which may be a macrophage or a dendritic cell, it digests the foreign organism and then separates the foreign protein or the antigen and then expresses it on the MHC protein. Now this MHC protein binds to a T cell receptor present on a T cell. In addition, both the antigen presenting cell and the T cell, they are also bound to each other by cell to cell adhesion proteins. Now we move on to the different types of T cells and their functions. The T cells are classified into three major groups: helper T cells, cytotoxic T cells, and the suppressor T cells. The helper T cells, they are again further subclassified into many other types like helper T helper 1, helper 2, and so on. In addition, these T lymphocytes can be classified according to CD classification. Now CD means cluster of differentiation, and these lymphocytes, they on their surface possess antigenic molecules which are identified by monoclonal antibodies and therefore numbered as CD1, CD2, CD3, and so on. Amongst these, CD4 T cells are the helper T cells. So another name for the helper T cells is the CD4 cells. Now these CD4 cells, they bind with MHC class 2 molecules as mentioned earlier, and they activate B lymphocytes to form the antibodies when the antigen is presented to them by the antigen presenting cells. Another important subtype is the CD8 T cells. They are basically immature T cells. They bind with MHC class 1 molecules and they differentiate into cytotoxic T cells. So the cytotoxic T cells, they are the CD8 cells. Now coming to the functions of the helper T cells. Secretion of lymphokines like interleukin 2, 3, 4, 5, 6, the granulocyte monocyte colony stimulating factor, interferon gamma. Another function of helper T cells is the stimulation of growth and proliferation of the cytotoxic T cells and the suppressor T cells. In the absence of the T helper cells, the clones for producing cytotoxic T cells and suppressor T cells are activated very slightly. Now, the lymphokine interleukin 2 produced by the helper T cells has a specially strong stimulatory effect in causing the growth and proliferation of both cytotoxic and suppressor T cells. Another very important function of the helper T cells is the stimulation of B cell growth and differentiation by the secretion of interleukins 4, 5, and 6. So, now these interleukins 4, 5, 6 have such a potent effect on the B cells that they have been called as the B cell stimulating factors or B cell growth factors. Another function of helper T cells is the activation of the macrophage system. Firstly, they slow or stop the migration of the macrophages after they have been chemotactically attracted into the inflamed area. And secondly, they activate the macrophages to cause far more efficient phagocytosis, allowing them to attack and destroy the invading bacteria or any other tissue destroying agents. In this figure, you can see the functions of the helper T cells acting on the B cells, the cytotoxic T cells, and the suppressor T cells. Cytotoxic T cells. The cytotoxic T cell is a direct attack cell that is capable of killing microorganisms and even some of the body's own cells. The receptor proteins on the surfaces of the cytotoxic cells cause them to bind tightly to these organisms or cells. After binding, the cytotoxic T cell secretes hole-forming proteins called perforins, special hole-forming proteins, perforins, that can cause holes in the membrane of the attacked cell. The cytotoxic T cell then releases cytotoxic substances directly into the attacked cell. Now, the cytotoxic T cells can kill the tissue cells infected by the viruses. In addition, the cytotoxic T cells also play an important role in destroying the cancer cells and the heart transplant cells. In this figure, you can see the direct destruction of an invading cell by a sensitized lymphocyte. Suppressor T cells. Suppressor T cells suppress the functions of both cytotoxic and helper T cells. The suppressor cells are classified along with the helper T cells as the regulatory T cells. So remember, when the regulatory T cells are mentioned, it means the suppressor as well as the helper T cells. The suppressor T cells play an important role in limiting the ability of the immune system to attack a person's own body tissues, a phenomena which is known as the immune tolerance. Now, I mentioned this immune tolerance when we were studying the pre-processing of the lymphocytes, and we know that the process of acquired immunity would destroy the individual's own body proteins if a person became immune to his or her own tissues. Now, the immune mechanism normally recognizes a person's own tissues as being different from the bacteria or viruses, and the lymphocytes which react against person's own body tissues are eliminated during pre-processing. Now, tolerance results from clone selection during pre-processing in the thymus and bone marrow. The failure of the tolerance mechanism can cause autoimmune diseases, and there have been many theories about the autoimmune diseases. One of them is that the destruction of body's own tissues releases self-antigens that can cause acquired immunity in the form of either activated T cells or antibodies. Immunization. Immunization has been used for many years to produce acquired immunity against specific diseases. Now, there are many methods by which immunization can be achieved. For example, immunization by injecting the dead organisms. Now, a person can be immunized by injecting dead organisms that are no longer capable of causing the disease, but still have some of their chemical antigens preserved, which can lead to the activation of the immune system. Now, this type of immunization is used to protect against the typhoid fever, whooping cough, and diphtheria. Another method is immunization by injecting treated toxins. And this type of immunization is used to protect against the tetanus and the botulism. And finally, a person can be immunized by being infected with live organisms that have been attenuated. That is, these organisms either have been grown in special culture media or have been passed through a series of animals until they have mutated enough that they will not cause disease but do still carry specific antigens required for immunization. Now, this type of immunization is used to protect against poliomyelitis, measles, yellow fever, and smallpox. I stop here to end today's lecture. We will continue with the rest of the topics in the next lecture.